Thank you very much for the invitation.
Please stay there because you are in the...
Yes.
Sorry.
Thank you very much for the invitation. And today I would like to talk a little bit about some biological problem associated with some computational problems. We do both experiments and computations, and most of our targets are transmembrane proteins. No, it doesn't. Are transmembrane proteins. Okay. So most of them are transmembrane proteins sitting in the cell membrane and use the energy from ATP to transport several different types of molecules like hormones. And some of them take part in recognition of toxic molecules, so they defend our cells have harmful molecules. And they are transmembrane proteins, so they are sitting in a lipid bilayer. And what is very important, that a significant portion of our proteins, approximately 25%, are transmembrane proteins. And if you go to the drugstore, then most of the drugs on the shelf are targeting transmembrane proteins. And in order to design drugs for curing some disease where the transmembrane protein is working inappropriately, you should have the structure of the protein at the atomic level. However, transmembrane proteins resist to structure determination. In the PDB structure database, there are over 100,000 experimentally sourced structures, but only less than 6,000 is for transmembrane proteins. A couple of years ago, Anfinsen proved that the sequence of the protein determines its structure. So we have 20 letters, 20 amino acids. We have very long words, and these determines the structure, the functional form of the protein. That was an experimental proof worth for a Nobel Prize. And from that time, computational biologists or computational scientists try to figure out how to predict the structure based on sequence. And that was really a hard problem. And actually, even a few years ago, we thought it will not be solved in a decade or more. But 2 years ago, a company of Google, DeepMind, appeared in the structured prediction competitions with a deep learning model. And this deep learning model became the best 2 years ago, and the model was released this summer. And this model basically takes 3 different parts. The first part is generating the input for the deep learning models, and they use the input structures of proteins which are homologous to our target sequence. I think it is relatively trivial. I think it's relatively trivial to imagine that if you have a structure which has a sequence very similar to your target sequence, that you can gain information from it, which amino acids might be close to each other. However, the other input, which is the sequence alignment of sequences which are homologous to your target sequence. And I have to tell you that it seems that this input has a much higher weight compared to the input structures. And in the next figure, I will explain how the information from the sequence alignment can provide information for the three-dimensional structure. You have a sequence alignment of homologous proteins, and if you observe that at one position, a large amino acid is maintained, replaced to smaller amino acids, and you observe at the same time that another position, a smaller amino acid is replaced to a larger one, so the opposite, I might have mixed, then you can make the inference that these positions are close to each other in space, since if there is a larger amino acid at one position, but you don't replace the other one, they will clash. So basically, the central block, the deep learning module of AlphaFold, extract this information, distance information from the sequence alignment. They use transformers with attention mechanism and a couple of small tuning. And at the end, what they do, that you collected a lot of distance pairs between amino acids, and this can be used to build a three-dimensional structure. You constrain the distances between pairwise amino acids with these distances, and you will have a three-dimensional structure. You will not have five, since they train the 5 different models. They also developed scoring functions, not scoring functions, scores, to tell something about the quality of the prediction. They use the so-called PLDDT score, so predicted local distance dissimilarity test. And if it is high, then the prediction is good. If it's low, probably it's not so good. And first, I will talk about the hardware background for this calculation, since it's pretty interesting. So the sequence searches to generate the alignments, I think it is the bottleneck. Not the disk space which is required. It's relatively large, but nowadays it's not a problem. However, it's IO heavy. So it's difficult to say, but let's say if your protein sequence is 1000 amino acid, which is not very large, it can go higher. This part can take 7-8 hours calculations. Some folks try to shrink the required database to smaller and keep it in the RAM, the memory. That might help, but you may lose precision because of the smaller database you use. I think it will be okay, just it's still difficult to access such a computer.
Sorry, what do you mean on reducing? So what you can, I have no idea what is the not necessary.
In the database, in the sequence database, you have almost 300 millions of sequences. And you already shrink these to have a 3 terabyte or 3 terabyte database. But they say if we decrease the number of the sequences, we still will have good alignment. In the alignment, usually I would say we don't have more than 10,000 sequences. And the quality means that if you can collect from the sequence database a good set of sequences of the prediction. It's also very interesting that I haven't checked that shallow alignments, 1-200 sequences might be enough. And that one can be selected from a much smaller sequence database. But it wasn't tested rigorously at this moment. But I think it will work. But okay, DeepMind also told that they used a computer with 85 gigabyte RAMs. I think that is really needed. On the other hand, if you have a very conserved sequence and you collect a lot of sequences, then you might have a problem, since the output of the sequence alignment is, the result is a file on the disk, and it has to be parsed. But if it's too large and it is parsed in the memory, then you are in a big problem. Yes, you have to correct the code. The second part, the running of the deep learning model, of course, you need a GPU. And for this, you also need some higher-end GPUs. It might be possible, I mean, most likely possible to use multi-GPUs. I haven't tested yet, since at this moment this memory was sufficient for me. On the other hand, I also ran into a problem in some of my simulations stopped with the error caused by TensorFlow. It has a hard-coded limit for a certain database data object, 2 GB, and fortunately, it was a bug in the AlphaFold code. As a biologist, I'm pretty proud to found that spot and insert 2 small quotes offline and eliminate this problem, and that was already included in the last release. So generally, I would say that AlphaFold DeepMind released not only the AlphaFold code, but they also released the prediction of protein structures for proteins of 22 organisms. This is over 300,000 structures. And DeepMind plans to release the structure for all proteins in 2-3 months, which means millions of protein structures. This really will change the paradigm of structural biology, not only computational, but also experimental, drug design, etc. But now I turn a little bit to biology. As I mentioned, membrane proteins, transmembrane proteins resist to both experimental and computational methods. And AlphaFold was not tuned for transmembrane proteins. They were part of the training set, I mean, some of them, but no special attention. So it's a big question whether the transmembrane protein prediction by AlphaFold is good or not. So first, you can take the structures deposited by DeepMind. You can select the transmembrane proteins. You can select the soluble proteins and check the distribution of the PLDT score, the quality score. And PLDT scores higher than 70 can be considered as good predictions. So as you see, similar amount of structures, similar percentage of structures of membrane proteins were predicted as good as soluble proteins. However, it's very important to know that this is just a score, and it also could happen that AlphaFold folds the protein, the membrane protein, in a manner which is globular, but the hydrophobic parts are inside the protein, and the prediction score is good. So you want to get a picture whether the AlphaFold predicted structures look like a membrane protein. And you can see the transmembrane helices, and if you place transmembrane helices next to each other, then the ends should be parallel at a certain level. And if you calculate the distance between the 2 ends, the set of the 2 ends, the distance of center of mass at the extracellular and intracellular end, then it should be somewhere 20-30 amino acid, because the hydrophobic region of a general membrane is so thick. And of course, if the prediction is wrong, for example, one of the helices is not inserted, then the center of mass in this case just get very close to each other, the distance. And we calculated first the distances for experimental structures, and they really fit between 20 and 35 angstrom. And as you see, for the AlphaFold predicted structures, most of the structures exhibit orientation of helices, which corresponds to a membrane bilayer or something like that. You might notice that here is a significant portion of predictions which seems to be not good. However, most likely one portion of this portion is really a bad AlphaFold prediction. On the other hand, I also have to note that we don't exactly know the location of the membrane lipid molecules around the protein. It's also a prediction. And it also could happen that in some of these cases, we couldn't predict the location of the transmembrane helices, but the AlphaFold prediction was good. This is somewhere a higher resolution information on the structure, but we still don't know how accurately the alpha helices are located next to each other in the protein. And therefore, we took a couple of transmembrane folds, transmembrane structures from our ABC proteins. It's a very diverse family. You can see that 8 very different folds exist in this family. And you can take all the ABC protein structures from the AlphaFold predictions, and you can compare each of the structures to every these experimental folds. You could calculate root mean square deviation, but there is a better score in the template modeling score, which can take values from 0 to 1. And if the value is above 0.5, that the two folds can be considered the same. If the value is below 0.3, then they folded differently. As you see, all of the AlphaFold predicted structures seem to ABC protein structures seems to have good fold. However, this is not a real proof that AlphaFold works well, since first of all, these structures and almost all of these structures were in the training set. Second, even if they were not in the training set, since their structure, let's say, was determined after 1918, April of 18. So on the other hand, when they deposited the predicted structure and when they predicted the structure, they also used available templates, even from 2020. So it's very difficult. I mean, you cannot judge the efficiency of AlphaFold on transmembrane proteins, also not on soluble proteins. But if you look for structures which haven't been determined before 18, or a similar protein structure were not determined, then you can use that protein for testing the AlphaFold. Tell you the truth, we found only 3 proteins, 3 structures, which have never seen by AlphaFold. 2 of them were predicted very well, just like the experimental structure. And this one exhibited some problem. And the model looked like this. You could claim that the protein is flexible and this is a different conformation, but if you look, the protein is colored from blue to red from the N-terminal to the C-terminal end, and the N-terminus is on that side, while in the other protein is this side, and it cannot happen. The membrane topology should be correct. Hydrophobic regions cannot really cross the membrane. So what we did, we just performed the experiment as long as we got the right structure. So several times with different random seeds. And here you see the PM score. Basically, all the predictions exhibited relatively high PM score, so not really below 0.5, but exhibited a couple of pretty high PLDDT score. Because since this is just one protein, I cannot state that don't use model 5, out of the 5 deep learning models for transmembrane proteins, but I'm getting suspicious. On the other hand, what is very important, the red dots and numbers indicate the top scored predictions from each run. So each run you get 5 structures, one of the top prediction colored red, and from 6 runs, from 6 top ones, there is one which basically exhibit the very same structure. And I think I'm over the time, isn't it?
You still.
Still, you know, I was written to have 40 minutes, but I don't plan. So usually, our main objective is to characterize how the drugs get into the protein, the pathway inside the cell, and how they are transported to the extracellular part. For this, we perform molecular dynamics simulations. And in the last year, we studied a protein from a plant, no structure, but I have built a lot of homology model based on homologous protein structures and sequences. So I also use the homology model for this study. On the other hand, this is already the AlphaFold structure, and this is the AlphaFold structure and the homology model overlaid. And the big problem we had that our homology model was not stable in simulation. This is characterized by the root mean square deviation from the initial structure. It was pretty high for the homology model, pretty low for the AlphaFold model. Actually, this is not so bad, but our main problem was that here in the middle, at the central drug binding pocket, one of the alpha helices got unfolded in the homology model, while it remained stable in the AlphaFold model. And the spheres shows that basically the location of the amino acids were good, no shift in the alignment. So the homology model was great, but the side chain packing was different, and that was enough to provide a stable helix. So we really were surprised that even at atomic level, AlphaFold can outperform other existing methods. So in general, I think that AlphaFold works as good for transmembrane proteins as for soluble proteins. It's very important that people are most interested at this moment in the structure of protein complexes. AlphaFold seems to work for complex modeling also pretty well, since if you imagine that you have a protein with different domains which are interacting in the learning set, that it's intramolecular interactions between different parts of the protein, then this is very similar than interactions between different proteins. However, DeepMind also trained a specific model for complex structure prediction. It was released not this Tuesday, last Tuesday. but it's they are doing pretty fast. There are also some drawbacks. The conformation is random. What does it mean? If I have two different conformations for the transport, open inside to the cell, so the substrate can bind, then I ATP change the conformation, and you see it's open to outside. And we don't know how AlphaFold produce which conformation. Sometimes I would need the outward open conformation, but I just get the inward open conformation. Yes, I haven't tried the AlphaFold multimer, but I'm pretty sure that I will run into memory problems, and I really don't like. I mean, I was pretty surprised. First of all, they had some bugs, and pretty important bugs. On the other hand, it was just so good to read their code compared to the code of other scientists. People like to say and state that the protein folding problem is solved. Protein folding is you have the straight peptide chain and you fold it to the three-dimensional structure. I would like to emphasize that not the folding problem is solved, but the structure prediction based on sequence. At this moment, we still don't know the important steps of the folding, which are very, very important in certain diseases. But I think that AlphaFold also helps in this. Okay, I really thankful to my group, to Gina and Bianca. But at least Gina just delivered a baby in April, and they made some fancies. They called the girl Regina, so Regina. Bianca got married 2-3 weeks ago. So, but hopefully now I have new computational colleagues, and we will advance. And I really would like to emphasize that I got a lot of help and input from a lot of computational people, hardware and ideas from Gergely group, and also from another ones like Max Planck Institute, and also sometimes Kifu helps me a lot. But of course, the current cluster cannot be used for this, but as you heard in the morning, hopefully the next one will be really powerful. Thank you very much for your attention. If you have time that we can have some questions, answers.
Thank you very much.